All right, so since we just worked through elongation, we'll just do a brief overview of what that looks like again, just to make sure everybody's on the same page. So, elongation is going to be step number 3 in the process of translation. Step number 1 was tRNA charging. Step 2 was initiation, where all the factors came together and assembled on the ribosome. And then step 3 was elongation, and this is where the assembled ribosome is going to occupy the messenger RNA strand and is going to start by bringing in the transfer RNA into the P site that is going to be, and that transfer RNA is going to have the initiating methionine associated with it. After the initiating methionine has come into place, then another anticodon region of a transfer RNA strand is going to occupy the A site, and then is going to bring in the charge transfer RNA, and then that charge or that new amino acid that's come into the A site is then going to see some energy exchange, and a peptide bond is going to be formed between the amino acids in the P and the A site. And then the transfer RNA in the P site is going to release its amino acid. And the ribosome will then move down the mRNA strand to the next codon, and we'll bring in another transfer RNA. And so at the end of the elongation process is where we see each cycle of elongation resulting in the amino acid that was in the A site being added to the polypeptide chain, and then the ribosome moving forward so that the A site can then accept another transfer RNA. So this is going to continue until the subunit actually comes to a stop codon. So keep in mind, when the ribosome actually reaches a stop codon, there's not going to be a transfer RNA unit actually that will code for that stop codon. What ends up happening is that once the subunit translocates to a stop codon and there's no transfer RNA that is going to pair with it in the A site, the release factor 1 is going to come in instead and is going to bind to the A site. And then release factor 3 is then going to form a complex with guanine triphosphate and is going to bind to the ribosome. Once this binding has taken place, the polypeptide chain is going to be released. And once the polypeptide chain has been released from the transfer RNA in the P site, guanine triphosphate is that was associated with the release factor 3 is going to be hydrolyzed to GDP, or guanine diphosphate, and the transfer RNA, mRNA strand, and release factors are going to be all released from the ribosome. So termination happens really, really quickly. Just keep in mind that there's actually going to be release factors instead of actual transfer RNAs that are going to occupy the stop site. So as we are wrapping up chapter 15, I just want to make sure that we go back through and actually talk through translation again, since it's kind of broke up among several online lectures. So when we go back to the beginning of this process, keep in mind that translation is really the second big part of protein synthesis. The first big part is transcription, which we learned about in chapter 13, and then comes translation, where we're taking the messenger RNA strand and translating it into protein sequence. So here, this process is going to first begin with transfer RNA charging, where the transfer RNA is going to be bound to an amino acid. We're going to see an exchange of energy happening, and that transfer RNA at the end of this charging process is going to be ready to be accepted by the ribosome. But before that can happen, initiation has to take place. So that's the second major step of translation. Initiation is where we see several factors come together. First of all, the small subunit is going to come together with the aid of initiation factors. It's going to bind to the messenger RNA, and that binding process is going to be very specific in terms of having the correct Shine-Dalgarno sequence for the specific gene that is being translated. Once the small subunit is in place and we get the release of some initiation factors, some conversion of some energy, then the larger subunit is then going to come in, set down on top of the messenger RNA strand, and is then going to be accepting the transfer RNAs that are going to be in the sequence after the initiating methionine. So during the initiation process, again, we have the small subunit binding to the messenger RNA with the aid of release or of initiation factors, the initiating methionine transfer RNA coming in, finding its anticodon sequence, and then the release of initiation factors and conversion of energy, where we get the larger subunit coming in and setting down on top of the transfer RNA. That signifies the end of initiation, and then we move into elongation. Elongation is going to be characterized as the ribosome has 3 different spots associated with it. So moving from right to left is going to be the A site, the P site in the center, and then the E site at the very end. The E site is going to be the exit site. So as elongation happens, the new charged transfer RNA that is associated with the codon of the messenger RNA is going to come in, associate with the A site. We're going to get bonding taking place. That peptide bond is going to connect the amino acid of the transfer RNA that is in the A site to the one in the P site. And then the ribosome is going to continue to move towards the 3' end of the messenger RNA. And that messenger RNA is then going to continue in this continue to be translated in this fashion as the ribosome moves from 5' to 3'. Finally, at the end of elongation begins the termination process, and the termination is going to be signified when the messenger RNA strand has a stop codon at the end of the coding region. That stop codon doesn't have an amino acid that's actually associated with it or a transfer RNA that's associated with it or has an anticodon for it. Therefore, the release factor will actually come in, occupy the A site of the ribosome, and terminate the translation process. So after that termination has been initiated by the release factor, the polypeptide chain is then going to be released from the last transfer RNA that's now occupying the P site, and then the subunits are going to disassociate, and the concluding product of this translation process is where we get that polypeptide chain that was newly translated from the messenger RNA strand. So table 15.4 in your book is a really good source of information to actually go through and look at the 4 different stages of this process, all of the enzymes and initiation factors and elongation factors that are involved with this process. And just keep in mind that we're going to concentrate on bacterial cells because eukaryotic cells get much more complex. There's much more regulation associated with it. So this is a really good table for you guys to start memorizing this process. All right, so I just want to make sure that we don't lose sight of what is actually happening in real time when it comes to translating messenger RNA. So keep in mind, especially in eukaryotic systems, that the entire genome different genes within the entire genome are being turned off and turned on at the same time. So those messenger RNA strands can actually be very, very, very long, especially with eukaryotic genes, like upwards of 10,000 base pairs or 20,000 base pairs long. So one of the ways that Mother Nature has really dealt with that issue is to have several ribosomes actually doing the translation process on the same strand of messenger RNA. And then in a post-translational model, those different polypeptide chains that are produced by each of the ribosomes will be packaged together by the Golgi apparatus. So here we can see up at the top our DNA strand. So we've got our leading strand and our lagging strand, and from that DNA strand during the transcription process, we're getting the production of messenger RNA. That messenger RNA strand can at the same time be going through the translation process. So the 3' end can still be getting going through the transcription process, and the 5' end of that mRNA strand can be starting the translation process. So this happens very, very quickly. And that that messenger RNA strand can have several ribosomes actually doing the work during the translation process, and that amino acid strand will be bound together in the Golgi apparatus. So this is what it looks like from an electron micrograph. So this is going to be the cellular material. Sorry, let me go back. And we've got an entire gene that is being translocated. That's going to be this line right here. It's very, very faint. And all these little tree branches coming off is going to be mRNA. And the round dot-looking things that are hovering around the mRNA, those are actually ribosomes that are synthesizing that polypeptide chain. So again, this is what it looks like. We get messenger RNA being translated in the 5' to 3' direction again. And once those polypeptide chains are free and floating in the cytoplasm of the cell, they're going to go to the Golgi apparatus and be packaged. And once they're packaged and shipped out of the cell, if that's what their function is, they can actually go through other modifications in different or at different points of where they work. So different points of contact. So some of the post-translational modifications that we see pretty often are like hydroxylation, methylation. This is a big one. We use it in agriculture all the time. Phosphorylation is a big one, and disulfide bonding as well. So we can actually see another layer of regulation of the genome and gene expression with post-translational modifications. If a protein isn't modified correctly after it's been translated, then it won't be active. All right, so that sums up post-translational modifications. I just want to talk very quickly about how antibiotics work because it relates to the translation process. So we know that when we get sick and have a bacterial infection, we can go to the doctor and get some sort of drugs. Well, those drugs are going to bind very selectively to the bacterial ribosomes and not allow translation to take place. So the bacteria tend to be different enough that the drug is going to bind them and not our own personal cells. And so antibiotics really prevents translation. So that's how we can get a hold on the bacterial process, and it won't let it go through the protein expression step as well. All right, so that is the end of chapter 15.